Today, we are going to talk a little bit about cell theory. Cell theory is extremely important to microbiology. We're going to talk about the basics of it and then also walk through a case study related to cell theory that might affect you. All right, so first of all, what is cell theory? Well, the idea of cell theory is two basic things. It's the idea that all cells come only from other cells, and that cells are the fundamental units of organism or the fundamental unit of life. Of course, you might wonder why this is important. Of course, all cells only come from other cells, except way back when, we couldn't even see cells because we didn't have microscopy, as I've discussed in previous videos. People didn't realize that cells had to come from other cells. They thought they could just appear magically out of midair. Some god put them there, or there was some life force running around the planet, and so cells had to come from something mystical or beyond them. In fact, one of the favorite things I learned back in microbiology was the idea that if you just threw a bunch of wet shirts over the hay in the barn for like a week, you would grow mice. It was a literal recipe for mice, and I think that's super interesting. But of course, now we know that cells come from other cells. Now, to learn that, we actually had to go through quite a process, and Pasteur, who you might know from milk pasteurization, is actually the scientist who eventually did the experiment that we now know of as the one that showed us that cells come from other cells. So he had this swan-necked flask. So it had this little loopy thing in it, and this thing does not want to draw the way I want it to, but it had a loop so that the microbes could not get into it. And then it had liquid in the bottom. So it had some kind of liquid-based stuff down here, liquid. And he would boil it. So Pasteur boiled it and then left it like this. Now, since it was open to the air, it should not have eliminated any life force or any kind of mystical energy, but any microbes that try to get in get stuck in this middle point, so nothing grew in the broth. And then there were some people who said, well, but hey, maybe you just destroyed the broth's original life force. But because of the way this flask was shaped, he was able to crack it open at that point and show that microbes could still grow in it. And then people said, all right, I guess that cells in the broth must have come from other cells in the air, and that's the way it works. And so then we have the idea that cells come from other cells. All right, but let's actually start with the idea that cells are the fundamental units of organisms. So what is that going to mean? If it's a fundamental unit of life, what are the things that we need to have a cell? What's its absolutely minimum capabilities and functions? And so therefore, what are the basic functions of cells and organisms? So what do you think that cells need to do to technically be alive? I think that there's really four basic things that if you don't have it all, then life doesn't really exist. And so every cell, no matter what kind, needs to be able to do these four things. All right, so what do we need to have a cell? First of all, since all cells come from other cells, cells need to be able to reproduce themselves. They need to be able to either split in some way to make more cells, have baby cells, whatever they do, they need to be able to reproduce from one cell to multiple cells, or at least make new cells. To do that, they're also going to need some kind of energy, so they need to have a metabolism. So metabolism is a generalized word that mostly is take in something, transform it into energy, and in some cases materials, and then spit out waste products. So every cell somehow needs to take in materials, create energy, and will spit out waste. Next, for a cell to be a living thing, it needs to respond to its environment. So it needs to somehow be able to take in information from around it and then also push out information or change things about its function to react to what's happening. Homeostasis is sort of a sub-level of responding to the environment because in the case of homeostasis, the cell's not only responding but also trying to maintain something internal. So it will adjust if something gets too high to bring it lower, and if something gets too low to bring it up. Either way, it's still a response. And then finally, the last thing you need to have some kind of living organism is evolution, which is change over time for survival purposes. So cells are going to need to change, and they need to be able to do so in such a way so that some die, some survive, and that's how you know that things are changing over time. So those are the four major things that cells need. We're going to get back to those at some point in the future, but it's still good to keep in mind the things we need to have a cell and what we expect this cell to be capable of doing. All right, let's move to the second part though. If cells only come from other cells. So a big part of microbiology, of course, is understanding the role of microbes in disease. So now, Pasteur did his experiment back in the 14 or 1500s. I can never remember exactly how old it is, but it's a while. And before that, people didn't understand that cells came from other cells. So they didn't understand that disease, especially bacterial disease, came from the outside world. They often thought it was a god or a curse or something they did to make it happen to them, but they couldn't understand how to stop it. But once you start to understand that cells come only from other cells, then it becomes easier to wrap your head around disease, because those cells had to come from someplace. So if somebody gets a cell inside their body, where did it come from? Did it come from touching another person or an animal? Did it come from the air? So what can you start to do? Well, if you know that cells come from somewhere, you can isolate people from those places. You can isolate people from each other. That was an early thing that people did way back before we even understood cells was, hey, if you don't put people who are sick near other people, they can't catch disease. So you get the idea of like leper colonies from the idea that if somebody had leprosy, Hansen's disease, which is caused by a mycobacterium, that they could pass it on to others, but if they stayed by themselves, other people wouldn't catch it. Now, of course, that was an extreme version of isolation, but now we know how to treat instead, so that's better. Another thing you can do, of course, is kill or destroy the cells that are getting in without actually fully isolating the person. So things like hand washing, which have actually only been a key part of medicine for a few hundred years now, came in because people went, hey, if these are cells coming off of a doctor and onto a patient, we can kill them just by cleaning the things we use to touch it. So cleaning hands, sterilizing instruments, things like that. Starting to wear things like masks to stop disease from traveling from one place to another. Switching up water such that you didn't use the same water over and over and you brought in clean water and got rid of wastewater. All of those things make a difference when you know that what's happening is a cell is coming from somewhere, getting into a body and growing into more cells. And then, of course, if you can kill those cells, you can stop disease, or at least in some cases. So all of that is extremely important in understanding how this process works. Okay, so now we're going to move forward and talk about a case study. So this is an end to ulcers, and it is a case study written by these two folks, Christy Hennin from SUNY Geneseo and Rod Hagley from the University of North Carolina, and I got this case study from the University of Buffalo Case Studies, if you're ever looking things up. All right. So, what do you think causes ulcers? This is something that has been pretty much passed down to us through sort of general thought processes, things that doctors have told us. These are all things that people definitely think relate to ulcers. Probably the biggest ones are the top two here, is that most people say, well, ulcers are because of stress, and the stress causes excessive acid, and because there's so much acid, it's clearly eating away at the stomach, thus causing an ulcer, which is an eating away of the stomach lining. But has that been studied? Do we know the answer to that? Well, let's think about it for a little bit. If we thought that acid was a main cause of ulcers, how would you design an experiment to test that? Well, if acid is the cause, we're going to have to think about how can we say, does somebody have acid or do they not? Now, there's a bunch of different ways to do studies. So one of the things you could do is you could examine people with ulcers and measure the amount of acid in their stomachs. Would that be sufficient? Probably not. You'd want to compare it to people without ulcers just to make sure, because stomach acid is very, very acidic. It's got an extremely low pH under normal circumstances, so it's a little hard to test the difference between someone who has an ulcer and someone who doesn't without actually doing both groups. Could you also try to lower the acid? That would be a great way to design an experiment. You could take people who have acid and have ulcers, take people with ulcers, but give them something to decrease the acid and see if it improves the ulcer. So that is a way to set up an experiment. So here's a study design that could exist. The group received antacids and could take them all day, and the other group received sugar pills, or a placebo. This is our scientific method, and took them. And then we assessed how many ulcers they had. So think for a moment about what the results would look like if the excess acid theory was supported. So remember, group 1 took the antacids. Group 2 took placebo. All right, so here's some possibilities. These are the different things that could happen. Now, in this one, both groups still have quite a lot of ulcers. So antacids, no antacids, doesn't matter. Ulcers all around, doesn't make a difference. In this one, they both don't have very many ulcers, in which case you got to wonder why you're doing this study at all. So A and B are really the ones we're interested in. So in group 1, it would mean that if you lowered acid, you had more ulcers than if you took a sugar pill and did not. That one probably doesn't make sense. But B would say, hey, if you took antacids, you had ulcers that decreased, and the other group with the sugar pills had ulcers at a higher level. So acid must be related. And wouldn't it be great if this was a seminal study in ulcer research? But unfortunately, interestingly enough, it was not a seminal study in ulcer research. We just kind of assumed it based on anecdotal evidence. So we had patients who took antacids, and it did decrease their ulcer symptoms. And if they stopped taking the antacids, their ulcers definitely returned. But it wasn't actually a controlled study. So from a scientific perspective, anecdotal evidence gets us somewhere, but it does not give us answers. And that's a really important difference. All right, so let's keep examining. What are the things that a doctor could do who wants to understand this? Well, they could look in the stomach. And in fact, that is what one of the doctors did. So in the early 1980s, Dr. J. Robin Warren, who was a pathologist, decided he wanted to examine stomach biopsies of patients with stomach ailments. And so he took them, he got some good cell samples and looked them over. You can see these are pretty much what stomach cells look like. They tend to be kind of squished together. Stomachs have to be able to expand and contract, so there's a lot of extraneous space. These are also fairly large cells because they are eukaryotic human cells. But inherently, it has a problem. He saw some smudges that he couldn't really see under low power. So he then said, well, what about a high power lens? What could I see at that point? So what's going to happen if I zoom in on this? Well, if I zoom in on this, obviously there's a bunch of different things you might see. But one of the important ones is that I can see much smaller cells. So eukaryotic cells are fairly big, but bacteria cells are very tiny. Am I going to get in close enough to see viruses? Almost definitely not. So if a virus is causing this problem, I'm not going to answer it with microscopy. But if a bacteria is, I got a shot. So this is what he did. He went under high power and said, oh man, these look a little bit like tiny rods, which should also look a little bit like some stuff y'all have been seeing in lab lately, tiny rods. And in fact, it wasn't a species that anybody had spotted before. They looked a little different, they stained a little bit funny, and so he said, well, this must be something completely new. And that's kind of exciting. And so they eventually named it Helicobacter pylori. Early on, it didn't have this name, but it did eventually. Although interestingly enough, even though he could get this image, the medical community didn't believe him. They were just like, that's crazy. This is utter nonsense. You can't possibly be finding bacteria in the stomach. Why? Why do you think his colleagues would be like, no, you can't possibly find bacteria in the stomach? Well, there's a bunch of possibilities. Bacteria are never found inside the human body. Although we've known for many, many years now that there are definitely bacteria in the gut and the mouth, so that's probably not it. But the pH is far too acidic for bacteria to survive. That one is pretty important because we did used to think that bacteria couldn't live in that acidic of an environment. Other people had not seen bacteria in their samples, although, again, he had to put it under high power, so that's going to make a difference. And then, of course, people just thought they knew. Ulcers and stomach cancer are simply caused by these things. Age, stress, diet. Bacteria clearly have nothing to do with it. The patients are here. We can tell that they're stressed. We can tell that they're older. We know they eat poorly. It can't possibly be a bacteria. But obviously there was a lot more going on here. And in fact, Dr. Warren went to the trouble of using a special stain. He absolutely made sure that it was impossible for these to be anything else, and his colleagues were convinced, which also actually got him a little extra help, and he was joined by another doctor. All right, but let's think for a moment. If we think H. pylori causes ulcers, so even if we believe there's bacteria, we say, yes, there's bacteria on this slide. Is that all that's happening? Bacteria are there, therefore they cause ulcers? Well, without some science, we're not going to know. So what would be another hypothesis? Think for a second. Why else could there be bacteria in these slides? And why might his colleagues go, nah, it can't be ulcer causing, it must be something else. Bet you could come up with some ideas. Well, here are the main ones. One, people would say the biopsy specimens were contaminated after samples were taken from the patients. People not using their good sterile technique. It's always possible for bacteria just to get into something. And okay, so maybe bacteria live in the stomach, but maybe they're always there. Always bacteria, always living there. They're in healthy people too. Maybe they don't do any damage. We know that's true of bacteria in other places in the body. And a third possibility be that the bacteria show up after there's already damage. So an ulcer occurs because of acid and stress. The stomach's defense is weakened, the wall is weakened, and the bacteria are opportunistic. They jump in and they go, hey, you made a home for me. And then they join up and are part of it, but they don't cause the ulcer. So all of those are possibilities, which of course we could figure out by doing a good controlled scientific study. All right, so Dr. Barry Marshall is going to join up with this research, which is fantastic. And they said, well, let's do at least a partial study here. Bacteria cause stomach ulcers. That's what we think is happening. So we need to do a study that's actually going to say, do the bacteria cause the ulcers? Obviously, this one is going to be tough. You got to do a number of different versions of the study. You can start with some basics. Let's make sure that if they cause ulcers, they're actually present. If they are not present in everybody with an ulcer, this hypothesis is going to go away immediately. So let's start with that. And they did indeed start with that. And they said, oh, hey, look, we took all of our ulcer patients, we took biopsies, we stained them, and they all have H. pylori. So that's a good starting point. But it does not eliminate any of the previous hypotheses. So to say that they cause it, we have to do one of two things. We have to either compare them directly to people who don't have ulcers, obviously, or do something that would get rid of the bacteria, but really change nothing else. If all you do is get rid of the bacteria, but you don't change the stress, you don't change the acid, you don't change anything, that will tell us a lot about what's happening here. And that's what they did. So they treated ulcer patients with antibiotics to see if the disease stopped. Is that sufficient? If we just treat all 100 of those people to see if the disease stops, then what? Oh, we are missing a key part of this study. Of all of these things, we have an experimental group because we're giving them antibiotics, but what are we comparing it to? You always have to have a group to compare, and that's your control. So be the control is the key concept. If you're interested in these other ones, our hypothesis obviously is on the previous slide. Independent and dependent variable are a little funky. Independent means that we get to control it. So it's whether or not they took an antibiotic or a placebo. And a dependent variable is dependent upon the study, so whether or not their ulcers got better. All right, so that takes us to what do we need to have a good experiment? A good testable hypothesis, bacteria cause ulcers, control and treatment groups, so something that changes and something that doesn't. And then we can actually do this study. All right, so we set up the antibiotic study. We have a treatment group. We've got a control group. In this case, our control is going to be here at the first bar, and our treatment's going to be in the second one. So remember our graphs from before. What might happen if? Okay, so first, what would happen if the bacteria thing is not true and it's actually still related to acid? So if it's still related to acid, would antibiotics make a difference? Well, if it's related only to acid, antibiotics shouldn't be changing acid at all, so they should not make a difference. So we can eliminate A and B. And if antibiotics were not making a difference, our patients would still have ulcers. So if acid were supported, we would see patients who have exactly the same number of ulcers, even with antibiotics. But if our new hypothesis that bacteria cause the ulcers was supported. Well, then we would expect our control group in A to continue to have ulcers. They'll have lots of ulcers still, but our experimental group, our group that received antibiotics, should lose their ulcers and become healthier. Is that what happened? Hey, look, it is in fact what happened. That's actually pretty cool. So when they were treated, 80% of the patients were permanently cured of their ulcers. They never got them back. So they didn't ask them to change anything else about their diet or anything. They never got them back. People still didn't believe this, which is kind of crazy. So Dr. Marshall, who didn't have ulcers, decided that he was going to do an actual cause and effect relationship because, again, people were like, well, maybe they're opportunistic. And so he swallowed the H. pylori. This is not a good idea. Do not drink any of the bacterial cultures in any lab that you are in. But it made for an interesting experiment because he did it, and within a week, he had gastritis and ulcers and was able to test and saw that H. pylori did indeed stick in his stomach. Then he was able to take antibiotics and show that all of his symptoms went away. Now that is interesting, and also it's a big part of what we would call germ theory. So cell theory, first of all, comes back into play because cells have to come from other cells. It means that to have Helicobacter pylori, you have to start with a cell and then it grows into more Helicobacter and then causes ulcers. If there are no cells, either because you don't start with them, so you never accidentally ingest them, or they are cured through antibiotics, so you destroy all the cells, they can't be there, they will not cause an ulcer. Germ theory is related, but a little bit different. So key concepts in germ theory involve the idea that an organism causes disease. You can isolate an organism from someone with a disease. So they did that in the early part of the study. They were able to say, here's Helicobacter pylori that we took from the stomach of someone with an ulcer. And if you take the bacteria out of someone with a disease and give it to someone who is healthy, they will then get the disease and have that bug. And Dr. Barry Marshall showed that. Now, normally, when we're trying to show germ theory, we don't give people the bugs. We track transfer. We see how they were in contact and if they could have picked it up from somewhere. And we do studies to check that the strains are the same from one person to another. We do not give people diseases for science. That's bad ethics. But in this instance, one person did it just to check it. And they were able to show the germ theory process, which is that the bug transferred, caused the disease, and when you killed the bug, the disease improved. So all of that relates to our understanding of microbiology and our understanding of disease and how the two relate to each other. So it actually was very difficult to get this information published. People thought they understood how ulcers worked. So they worked very hard to get this into papers. And actually the original paper that said, hey, we think there's bacteria in the stomach that might be a problem was published in 1984. And it took until the early 2000s for people to actually, for doctors and scientists to say, oh, maybe bacteria do cause ulcers, and for that to actually be part of the medical community. And we're in 2021 and people are still being told that when they have stomach issues, they should take antacids instead of being treated with antibiotics. So you can see how long it takes. Science changes at a pretty rapid pace. People don't change that fast. And so it can be very difficult even when we have good evidence to do this. However, in this instance, even though we still have some trouble in the medical community, the scientific community realized what had happened. And in 2005, they awarded Drs. Warren and Marshall the Nobel Prize in Medicine and Physiology for this discovery. And actually, it's gone forward for us to look at bacterial and viral causes of other things that we used to think were just age, stress, things we couldn't place. And there's been a lot of work done on human health to help out with that. So I hope you found that interesting and exciting and learned a little bit something and practiced your thinking about scientific method, microbiology, and cell theory. Thanks.